Welcome back to Biomindscape. This is a pull-down assay. Pull-down assay is an in vitro protein-protein interaction technique used to determine whether the two proteins physically bind to each other, and it is similar to the co-immunoprecipitation, but instead of using an antibody, it is used to target the bad protein attached to the bead to capture its interacting pre-protein. So a purified bait protein is fused with an affinity tag such as DST or his tag and immobilized on the affinity bead. So when a cell lysate or purified protein mix is added, the protein that specifically interact with the bait bind to it. So non-binding protein are washed away. The bound protein are evaluated and analyzed using by the SDS page or Western blotting. So first is the preparation of the bait protein. So the clone of the bait gene into an expression vector express the bait protein with an affinity tag like JSD, his tag or MBP tag. Then immobilization of the bait, that is DST tag, that is glutathione agarose bead. Then and his tag is the nickel NTA bead. So the bait protein become attached to the bead. Add to the prey protein, add a cell lysate or purified protein containing this suscepted interacting protein, incubate to allow the binding, then wash the bead several times to remove the protein that binds the non-specifically, and then release the bait prey complex from the bead, then analyze the protein by SDS-PH Western blotting or mass spectrometry. So there are two types: GST pull down, use GST tag bait protein, GST bind the glutathione bead, and his pull down use the his tag bait protein and his tag bind the NINTA bead. So what are the application? It detect the protein-protein interaction, validate the interaction predicted by the bioinformatics and study the signaling pathway, and in identifying binding the partners of newly discovered protein, drug target validation. And what are the advantages? It is simple and inexpensive, highly specific with proper control, does not require antibodies for the bait protein, can identify directly binding partners. So what are the limitations? It requires purified tag bait protein, may miss interaction that need other cellular component, weak or transit interaction may not be detected, non-specific binding can occur if washing is insufficient. So pull down assay detect the direct protein-protein interaction, GST tag bind the glutathione beads, the his tag bind to the NTA beads. It detect the common by it is detected by commonly by the SDS page and the Western blotting. Unlike the co-immunoprecipitation and pull down assay, use a tag bait protein instead of an antibody. Thanks for watching. Do like, share.